Okay, we're about 5 minutes after. Here's Alex jumping on too. So we're very happy today to have Neal Amin from Sergio Pasca's group talking about morphogens in vitro organoid systems to control neural fates, among other things, as we'll see. And before I introduce him formally and get him on to telling us what he's been looking at, I wanted to give a brief, just sort of probably what many of us on the call sort of are familiar with, but there's really an explosion, not only in organoid work, but immediately in the last couple of months on morphogen control of these in vitro systems, really to expand their study. So last session, we had Soraya Scuderi from Flora Vaccarino's group and Xiuchuan Lin from Barbara Trutline's group in organoids and the NGN2 transdifferentiation system, respectively. On this topic today, as I said, we have Neal Amin with a sort of an expansive study looking at a multiplex morphogen screens with organoids. And next session, which there's a date change, I'll send this out on an email because it coincides with the ISSCR meeting, actually. Two more talks from Barbara Trutline's group on morphogens from Akanksha Jain and Fatima Sanchez Calleja, who I believe is on the call, so welcome. But this is just to point out how much activity is here and how recent it is. Here's Soraya's paper on bioRxiv, which actually came out since she spoke to us. So feel free to look it up, or if it's on the seminar webpage, the link is there now. Xiuchuan Lin's paper is up on bioRxiv. Here's Neal's from today. And the next two talks from Barbara Trutline's group are also on bioRxiv. So all five of these papers, not only, they're super recent. This is all happening now. And in addition to this in vitro work exploring the function and molecular mechanisms underlying morphogen dynamics and regionalization of the CNS, in recent years, the in vivo work has gone kind of in the same way, a little bit before the in vitro work. The Mano and Linnerson in 2021 have a study in the early mouse that really detailed with sort of precise details about where and when these signals come up in the mouse. At the end of last year, I think it was October, Nicola Micali and Pashka Rokisha's group put out a developmental atlas of the macaque telencephalon, where they again really focused in on the different cell types and different organizers in the primate brain. And even more recently, or maybe it was actually at the same time as the Micali paper. Yeah, it's October of last year. Emily Brown and Sten Linnerson's group put out a paper on the first trimester human single cell data. And again, looking at these early organizer states. So there's a huge amount of activity here. There's the in vivo atlases. There's all this in vitro mechanistic work now being done. And on top of that, I think someone else who's on the call, this is now a throwback to 2015, Jessica Mariani in Flora Vaccarino's group, looking at autistic spectrum disorder. And this was one of the first observations that these regional decisions could be perturbed in human disease. Despite the fact, of course, that everyone with autism has a ventral and dorsal pallium, there is perturbations in how they proceed in vitro, at least, and potentially in vivo. And to now fast forward again, here's a bioRxiv paper about a month or two old, again in autism, from Dan Geschwin's lab, also involving Sergio Pasca, where they're studying 70 different lines. So there's sort of things are coming together here in a kind of incredible way between the developmental in vivo data, the in vitro morphogen regionalization data where we can get into mechanism, and then studying larger and larger numbers of individuals with normal individuals and with different psychiatric and neurological diagnoses. Very quickly, we're finding ourselves in a place where if we can harness all this data together, things become incredibly exciting. So I'm very excited to have Neal talking to us today and to have this whole series of morphogen-related in vitro studies in our series. And what I'm going to be trying to do between now and the next session in late July will be to help anyone interested. I'm already working with Soraya and also Alex Jordan, who has an autism study in organoids from the Vaccarino group, is to bring a lot of this data together. And I've sent this group a lot of emails about links to NEMO Analytics, where we can bring all this data together. And I'm really going to try to work with everybody and invite everyone to sort of join in this effort of bringing data sources, and even gene signatures. If you think you have a gene set that you understand does a particular thing that you'd like to look at across this data, it's just as interesting as having another data set here. But once we get all these things together and we can start to look at them together, I think we'll really start to be able to understand and see things. Given how much data is out there, it's really incredible. So with no further ado, I'm going to introduce Neal now, but just to let you know, I'll be trying to build these data resources and invite you all to jump in as much as you want. All of our speakers are, of course, welcome to get their data up on NEMO so that we can start to have a really sort of incredible data sandbox to look at all these incredible data resources together. So today, Dr. Neal Amin is speaking, generating human neural diversity with a multiplex morphogen screen in organoids, from Sergio Pasca's group, who's had, of course, a long history, or it's as long as you can say that organoids have existed, has a lot of work there. And the sort of volume and detail of work that Neal has put together here is really incredible. So I'm very glad to have the sort of guided tour today to get our heads wrapped around what he has seen here. Neal, thanks so much for coming, and you can take it away now.
All right, let's see if I can screen share here.
Great.
Is it coming through full screen?
And we have we see the presenter mode.
Presenter button. Okay, let me try this again. How about now?
Looking great.
Great. Okay. Well, thank you, Carla, for the introduction, all the work you're doing, putting all these recent papers together and finding commonalities, and also assuming also trying to think about what's coming next, what are the big questions that remain, how to strategize around taking new technology and stem cell biology to address questions we haven't really been able to answer before. So as mentioned, my name is Neal. I'm a clinical assistant professor at Stanford, working with Sergio Pasca. I did my PhD at the Salk Institute with Sam Pfaff, where I studied spinal cord development and motor neuron biology and post-transcriptional regulation in cell types in the spinal cord. And then here at Stanford, I finished my residency in psychiatry, and then I've been using stem cell models to model both human neurodevelopment and also develop them into models of disease. And what you can see here is some cleared organoids, and the way I see them is that they're, our goal with these is that they can encapsulate something about different regionality of the human nervous system. And as mentioned, this works on most of this works on bioRxiv. They'll present some work at the end that is unpublished that will go into our final publication. Okay, so it sounds like this audience has thought about this question a lot, but it's worth just reconsidering why exactly we need a human cellular model of the brain. So obviously there are just very big differences, and obviously here in size, but also in many other attributes as well. So some of the other key features that are different between human and other species is that, for example, for mouse, which has been a really important model system for a lot of neurodevelopment of mammalian systems, has diverged from human 100 million years ago. And that means there's 100 million years of acquired mutations and genomic rearrangements and changes in signaling pathways that enable, for example, just a larger nervous system, but also different structural features as well. On the top right, you can see that an example of how genetics of a major neuropsychiatric disorder don't really correlate well with models in rodents. The lissencephaly gene, LIS1 gene, when disrupted in humans, causes a really severe neurodevelopmental disorder where children are born with a very smooth brain, a lissencephalic brain. And you can see on the left side of that, the rodent brain is always lissencephalic. So when that gene was knocked out there, there's really no change in structural phenotypes, and it's nowhere near as severe as behavioral phenotypes is what happens in humans. And then on the bottom right is the result of data from single cell sequencing of different species in different brain regions. And in this work, this group had found a population of interneurons that they've identified as being primate specific that were not present in the mouse striatum. And these are marked by the expression of the TAC3 gene. So all of these features are just examples, but there are just many differences. And also when we think about modeling of human neuropsychiatric disorders and complex human neurophysiology, I think it's really important to consider using a human cellular model. So again, as many people here are already aware, morphogens are key factors in giving rise to cellular diversity, both in vitro and in vivo. So in this case, taking somatic cells, reprogramming them into human stem cells, that's really the first step. And then from there, we can obviously derive them into, or differentiate them into a wide variety of cell types from nervous system or muscle cells, fibroblasts, et cetera. And really this whole process relies on a relatively basic building block of signaling, which is that morphogen gradients are established in vivo. This is due to organizer cell types secreting molecules. And then cells that are receiving these signals have transcription factor networks that integrate these signals and cause them to differentiate into a discrete cell state. In some cases, they're not as discrete, but for example, in the spinal cord, you have different distinct spinal cord progenitor domains that can be arranged dorsoventrally. And as we were discussing before, I think many people joined the call, that this work is really grounded in work that happened many decades ago. This is just a couple of examples that of work that really established this basic principle that we're still using today. On the left is work from Drosophila, how groups were beginning to understand how a single morphogen, in this case the bicoid in Drosophila, could cause these distinct expression patterns spatially oriented in the developing embryo. And on the right side is pioneering work really establishing how what cell types are really creating these gradients. So the notochord is shown on the top panels. On the top left is a normal notochord position, just ventral to the developing neural tube. And on the right is the result of a graft of a notochord to the dorsal part of the neural tube. And you can see on the bottom panels the staining of spinal cord populations that this grafting of the notochord can induce motor neurons in the dorsal neural tube where they usually don't exist. And experiments like these really establish that it's secreted molecules from certain signaling centers throughout the developing nervous system that orient anterior posteriorly, dorsoventrally, all these different dimensions, and that receptor progenitors and cells can integrate these signals to decide what cell type they're going to become. Now, in isolation, each of these kind of one molecule at a time experiments can establish some of these principles, but there really is just an incredible amount of neural diversity in the human nervous system, in the mammalian nervous system, and our understanding of how these various morphogens really integrate to give rise to each of these populations and the precise patterns and precise locations in the brain is still not fully understood. And what I was interested in is with newer technologies like single cell sequencing and also now multiplex single cell sequencing that we used here, that we can start to treat this as a more of a mathematical, a technological question. And even in the future, this could be maybe even broken down more by expanding the number of conditions used and really treating this as a big computational problem. So what we have learned from previous studies is that the identity of the morphogen matters. The timing at which that morphogen is applied during development is quite important to its action. Also, we've learned that how long a progenitor has received a signal for, as the duration of its signaling, can impact its final output. And then also the temporal sequence of these morphogens, and also obviously the concentration, can all impact this process. And at the end of this, we get a nervous system that's full of neurons with different neurotransmitters, vulnerabilities to disease, connectivity, and so on. So here's the general schema of what our screen was designed, how our screen was designed, and what it was meant to do. So we use the organoid system that Sergio has spent the last 10 years developing. So first we take stem cells, we make them into spheroids. We then neuralize them with dual SMAD inhibition for 6 days. This allowed us to get really highly enriched population of neural ectodermal lineage cell types by that stage. And then from day 6 to day 21, we applied a variety of different morphogens listed below in different concentrations, timings, durations. And because organoids have obviously a 3D structure to them, and we add a morphogen, they don't reach all the cells equally. We wanted to make sure that organoids weren't going to fuse and change sizes during the duration of the screen. So in order to keep the organoids separate during the whole screen without them fusing, we generate this PLA grid using 3D printing, in which an organoid could go in each individual well, and this well would be placed in a 6-well dish, and we could still easily feed each of these. All the while, we could culture thousands of individual organoids separately. After day 21, we culture them for additional 50 days and then perform single-cell RNA sequencing, which with each condition individually barcoded using the Parse Biosciences kit, which is based off the split pool barcoding technology that was published maybe like 2019 or so. And we didn't want to just rely on single-cell RNA sequencing to validate our results. We also did organoid cytoarchitectural analyses, also did a lot of immunohistochemistry, qPCRs to get at what cell types we were getting. Okay, so as was mentioned, there are starting to be a wider number of single-cell atlases of the human developing nervous system. We were particularly interested by the Braun one that was mentioned from the Lenarsing group, because it was, first, it's one of the largest single-cell efforts across the developing human embryo at stages of development that are most appropriate for our organoid screen that's sequenced, I think, around 1.7 million individual cells. And then with that data set and our data set, we could integrate them into to see where our cells overlaid in this atlas. And you can see the results of that on the bottom left. In gray are the primary cells, and in black are our cells. And you can see the coverage of the spatial area of these populations that we were getting in our screen. And on the right side, you can see the different breakdown of different brain populations. And so we were satisfied that our goal was to really generate as much brain diversity as we can to deconvolute which morphogens were generating which cell types. And here we had coverage, I think roughly like 75% of their clusters. We had some of our cells in those. So then, just to really kind of summarize a lot of work done on the annotation side, and this is really something just important for any of the groups and all the groups doing single-cell RNA sequencing, is that the process of really finally annotating these populations is not automatic, is actually quite, we have to be very detailed and careful on what we're calling which population, often because a single single marker gene is really insufficient to mark a individual cell population. And so it takes a real a candidate-based effort and manual effort, and also can be used. We can also use label transfer methods computationally to get at what these labels are. But in this screen, you can see we've generated glial cells and neurons, really diversity of both populations, from choroid plexus to floor plate cells, thalamic progenitors, hindbrain progenitors. And on the neuron side, we generated telencephalic neurons, many interneuron populations, VGLU2 positive populations, and so on. So on the left is a pretty traditional UMAP where we've color-coded each population by its identity, cellular identity. But because we know where each condition each cell came from, we can also label each cell by its condition. And because we had so many conditions, we've summarized them here by grouping conditions that had similar molecules together into one color. And you can see how well these populations segregate by color here, and this is by the morphogen that they're exposed to. For example, on the oligodendrocyte precursor side and oligodendrocytes, you can see they're mostly blue. That means they're coming almost exclusively from the retinoic acid and sonic hedgehog treated population conditions. And also you can see up top for the choroid plexus and ependymal lineage cells, most of them are coming from the Wnt activator KIR with BMP together. In this way, we can look at both cellular identity, but also in which morphogens were applied to each of those. We want to go further and really try to integrate these types of data in new ways, because this was new for our lab, is doing multiplex single cell. And so this figure on the right is really an attempt to create a morphogen map of how to get a specific cell type and what morphogens were applied to get there. The way we got there is we took each individual condition, and you can see on the left, you can see where some of these conditions would map to on this UMAP, and then calculate the percentage of cells that we're mapping to each neural cluster. And so on the top, you can see in each column are the neural cluster numbers. And on the rows, you can see the molecular conditions, each molecular condition, how much each condition contributed to each neural cluster. So just as an example, we can take, let's say at the bottom, these bottom conditions with BMP7 and KIR applied early. So this is before day 13. You can see the conditions labeled on the far right side, so KIR at 3 and lower concentration, so on and so forth. And then you can see which neural clusters these organoids generated. And so you can see most of the clusters did not have a contribution from these conditions, meaning that each organoid is actually very specific into the cell types it generated. We didn't have any organoid conditions that generated like a massive diversity within one condition. And that was our goal, to kind of focus these protocols down specific lineages. And you can see for this cluster, this grouping here, these are Calretzius and cortical hem populations. So beyond the single cell data, we also wanted to track other features of neural development, such as structural and size features, because these morphogens don't just impact cell differentiation, but also proliferation and cell structure too. To look at that, we used tissue clearing and also morphological tracing over the 70 days of culture of these organoids. And you can see these are just the two dramatic examples, that FGF8 in the culture versus retinoic acid in the culture really dramatically changed organoid size and also internal structure here. And you can see more clearly on the SOX2 staining that the top one had many more ventricular-like zones than the retinoic acid one that was much smaller and had distributed SOX2 positive cells. What we also did to, what I was really wondering about with single cell technologies, and I hadn't really seen, is how much can we trust the quantification of a certain cell population by single cell versus what we might do for a traditional method like immunohistochemistry. So what we did was we chose a single transcription factor with really high antibody staining and quantify them in every single condition across multiple organoids, and then compare them to the amount of olig-2 that was seen in single cell RNA sequencing. And you can see the results of that plot here, where each condition, the organoids from each condition are plotted here. On the x-axis is the olig-2 percentage by staining, and on the y-axis by single cell RNA sequencing. And you see the high-level correlation here in the roughly a slope of 1, suggesting that the single cell data is actually quite useful and quite accurate in compared to other methods. And of course, none of these are, absolutely true. We never can truly know unless we were to somehow count every individual cell in an entire organoid, but they're still correlating quite well. And then we also track sizes of the organoids over time. And a nice sanity check was that if we added high concentrations of growth factors, the organoids did indeed grow a lot faster and larger overall. And we did see somewhat of a plateau after day 50, where organoids stopped growing in size. And from our screen, there's only one condition that we weren't able to sequence, and that was the one that was treated with BMP4. And you can see that that size of the organoid decreased progressively over time, and we believe that's likely due to differentiation of cells into the neural crest lineage. And those cells, when they're in the organoids, they kind of slough off from the organoid, and the organoid loses its structure because most of the cells are these usually very highly migratory cell populations. But we didn't exclude any of the other conditions other than that one. So because we have all this data from morphology that we could quantify and cell populations, we can start doing more interesting kinds of correlation analyses to see how these features might interact. And so what we did is do a multivariate correlation matrix. What this means is we're taking each condition, which has certain quantified morphological features and contribution to different neural clusters and cell fates, and start running correlations across all these conditions to see which morphological features correlate with different regional identities. So you can see on the top is the different morphological features we're putting in here. So olig-2 by immunohistochemistry, roundness, which was done in Fiji, ventricular-like zone counts, and then size at various time points. And so you can see here at the top for cerebellar so this is organoids that were generating cerebellar cell types tend to be much larger in size, and that's this positive correlation here, but didn't seem to have many more ventricular-like zones than other conditions. For example, the telencephalic population had many, or was very highly correlated with the generation of ventricular-like zones, but overall actually didn't have a much larger size. So this means that ventricular-like zones aren't really necessarily determinants of large organoid size. And on other populations, say the oligodendrocyte lineage, as we'd expect, had a lot of olig-2 positive cells in the immunohistochemistry. And then these were much smaller in size compared to the other organoids. So focusing on just a couple of interesting things that we saw on our screen was that it was the addition of FGF2, but not FGF4 or 8, that generated cerebellar neurons. And just as a note, in our screen, we added the N2 supplement, which contains insulin. And it was previously known that actually that FGF2 and insulin together could generate cerebellar populations. In our screen, we actually had them staggered so that in the first 21 days, there was no insulin applied, and it was only FGF2. Only after day 21 was insulin applied through the N2 supplement, suggesting that cerebellar cell fate specification can actually occur based on signaling of insulin even after the day 20 time point. Something else that came out of this that was interesting is that these, I mentioned before, timing windows can be really important for morphogen action. We found that the Wnt activator KIR, when applied early, so this is either from day 6 to 11 or day 11 to day 16, resulted in the generation of cortical hem and Calabresius cells. But if the Wnt activators added after day 16, it seemed to have no major effect on cell fate specification. And these cells were really no different than ones that didn't receive any Wnt activation at all. And they generate telencephalic progenitors and VGLUT1/2 positive telencephalic neurons. So that means there's seems a really specific and really fine-tuned switch in Wnt's ability to drive cell fate in the stem cell model right around day 16. So we were curious in our screen. We were generating lots of populations of large clusters of neurons, but we're also curious if there are any smaller populations of neurons that we might be generating from our screen that might be relevant or interesting too, and what really is the resolution of a screen like this. And so one of the conditions in our screen generated these NKX2 positive interneurons that also seem to have a expression of the TAC3 gene that was recently seen to be a marker of these primate-specific interneurons that were recently discovered. And so on our screen, we saw that high activation of the sonic hedgehog pathway and medium activation of the Wnt pathway could generate roughly 5% of these TAC3 interneurons. Judging from our other conditions, we came up with a hypothesis that maybe too high KIR of Wnt activation is not going to generate these, but maybe we could push the sonic hedgehog activation even higher, and we might get more of them. So to test this and also validate if these are indeed the same population of TAC3 positive interneurons I previously identified, we dissected out the developing human striatum and performed single-cell RNA sequencing. And again, we identified a transcriptomically similar population of NKX positive TAC3 interneurons here, validated them with RNA-scope, and then also provided another differentiation with multiple levels of sonic hedgehog, and found that we generated the most amount of these TAC3 interneurons when we had the most amount of sonic hedgehog, even more than our previous screen. And so this showed us that indeed we could reproduce the results from our original screen, even for rare populations that were only at 5% of a single condition, and that we could use that to generate new hypotheses that maybe even more sonic hedgehog would generate more of these more of the cell type, which we did. Okay, so this is getting into some of our newer work. So we don't have a very interactive ability here, but I want you all to when you think of the most beautiful neuron in the brain, you can shout it out or to yourself. But we can also now figure out the correct answer, because this is now crowdsourced across all of large language models. So whatever ChatGPT tells us is the objective answer. And so that's the Purkinje neuron in the cerebellum. And so we wanted to try to get at the most beautiful neuron in the brain. But we hit a snag that I think many other groups have hit too. So on the left is Ramon Cajal's is like the coffee table book that came out several years ago showing the Purkinje neuron. And what we saw in our cultures was that we were waiting and waiting and waiting for something like the left to appear. And after 8 months in culture, this is what I was still seeing. And from previous reports too, after many months in culture, they don't seem to be generating the dendritic complexity that we would expect. And this isn't just specific to Purkinje neurons. This is true for other types of neurons too, that we can generate these populations in vitro, but that there seems to be a maturation wall that's being hit, either due to nutrient inavailability, some other kinds of homeostasis that's controlled for in vivo versus in vitro, or something else that we don't know about. So we wanted to try to address this question of cellular maturation with the Purkinje neuron by transplanting organoids from, this is the protocol from our screen, the cerebellar organoid protocol from our screen, into the rat cerebellum and observe its maturation over time and track the maturation of these human Purkinje neurons in vivo. As you can see, the location of our implantations on the left, and we track the growth of these organoids over time. And here you can see the rat cerebellum and the human cerebellar organoid now grafted into the rat after couple hundred days of development. So a few things that we saw that were really surprising. One is that Purkinje neurons apparently really like to migrate out of the graft. We saw this even after maybe 50, 60 days after implantation. We had sacrificed one animal just to see what was going on. And the farthest cells that were growing outside of and migrating out of the graft were always Purkinje neurons, which was surprising because at least for me, I didn't really expect that to be the case. And more interestingly, when they also seem to form these clusters of neurons together, and also have this orientation of their dendrites, as you would expect in the rat, in the normal cerebellum, where dendrites are positioned in the same directions and axons are projecting out the other direction here. And you can see a zoom in of some of these dendrite processes here. So Carla had mentioned Pascal Rakish is still publishing groundbreaking work even now. And what was interesting to us is when I tried looking at any reference for human Purkinje neuron morphology development, I couldn't find anything since 1976. And this is Pascal Rakish's work from back then, where he morphologically tracked the development of human Purkinje neurons. And this is the only way you could accurately stage these neurons that we're developing both in vitro and in the transplanted organoids. And in vitro, you can see they have this bifurcated morphology, which is in the fusiform stage before we get to the complex branching stage, which is more characteristic of mid to late human neurodevelopment. And you can see from our transplanted as THCBOs, they generate immensely more dendritic branching, complexity. You'll see the orientation of dendrites in one direction and the axons going the other direction, and we can quantify that here in this shoal analysis. So what I've shown you here, this is a summary of the work, is that we take a single cell multiplex organoid screen of morphogen action, can integrate this into a brain morphogenic map of how we can use different morphogens to generate specific cell types. We generate new protocols for certain cell populations. We hit a wall with maturation of these organoids, but we were able to get past that by transplantation, most exemplified by this by the Purkinje neuron maturation. And then we've also generated a primate-specific population of interneurons that was recently described using human stem cells. And I think these technologies are really impacting many different areas of biology. And I think with all of this, it's really important, in my opinion, to not treat it purely as a technology, but really see what questions can really be answered with them that could advance our understanding of human neurodevelopment and disease. So with that, I want to thank everybody for Carla for inviting me, and then everyone for listening in. This was done in the Pascal lab here at Stanford. My co-author Kevin Kelly, and then many people contributed to this work in very different ways. My funding sources are in the BBRF, the foundations of the National Institutes of Health, and the National Institute of Neurological Disease and Stroke with my KO8. And you can see this depth-coded, color-coded projection of the Purkinje neuron graft here. So with that, I will stop and I can answer any questions if anyone has any.
Thank you so much, Neal. It's amazing to see how much CNS real estate can be modeled. That's really a really cool ride you took us on there. Any questions, guys? This isn't something you mentioned, I don't think today, but sort of in the spirit of where we sort of started with developmental diversity. Do you have any insight into variability in these protocols across lines?
Sure, yeah, that is a great question that is going to be, I think, of increasing focus as I think many labs are trying to know how we can standardize all of our work. I mean, at least with mouse genetics, there are specific strains that some people use more often than others. And even in mouse genetics, there's just high interline variability. So it's not, I think it's comforting to know we're not alone here in stem cell biology of having a line variability issue. And I think we should have high standards, even though, I know definitely, having worked in mouse mouse modeling for my graduate school work, that most mouse behavior experiments are done in one line. I don't know many people who are doing it. And the ends are very, very small for some of these studies, because you have to train these animals so much that you can have an N of 2 or 3 even for a final figure of Cell Nature Science or something. So so this is a challenge for everybody, resource allocations and everything else too. So what we've done, see the cerebellar protocol in 3 independent lines. We've had actually, I think there are 2 other lines that we did in after the screen had even better hindbrain percentage than our initial screen. We've done the TAC3 line in multiple lines. We did our hypothalamic line with the best way we could assess that was with qPCR, because a lot of the transcription factor markers they don't have good antibodies for them. But we did that in, I think, 5 lines you can see that in our supplement. And then also with the Calvert-Cesar line, we did that in 3 lines as well. And so I think the main source of one is line variability, but also, these are tricky protocols. You have to feed them at, for 20 days, that critical period. If you're, out on vacation for a weekend, someone else to feed your cultures, or you feed them in the morning versus the evening. We don't know how much of these things impact things. We do know that even within the same line, we can, do the differentiation again, and sometimes you don't get as many of the same population as we got the first time. And so I don't know how much of overall variability in all of in the whole field studies is due to just that feature, which is just hard to do the same thing every time, versus the line variability. I bet it's a mixture of both. And yeah, how to work with that is something I think I'm sure everybody's thinking about ways to try to address that, but my guess is as good as everyone else's.
We have a couple more hands raised here. Alex, you want to jump in and ask the question?
Yeah, sure. Hi. I don't know if you can hear me correctly.
Yep, we got you.
Yes.
Yeah, good. Sorry, I'm on my phone. Yeah, no, great talk. It's really impressive. You're really starting to develop like the full recipe to get any kind of brain regions. I was wondering if you got like you could achieve to get the same multiple path. Did you observe like different combination of morphogens that generate the same lineage in your study?
Yeah. So I'll just bring this up really quickly again and see. Yeah, I think we did see that happen. For example, these Calvert-Cesar and cortical hemp populations, we got there by Wnt activation with KIR, either high or low. With higher amounts of KIR, we did see that we maybe got some more of these mice to forebrain interneurons. And then also got them with BMP7 and also with early KIR. So, I think the 2 ways of seeing all this stuff is in a reductionist way. It's like, what is the minimal amount of anything that you would need to get this population? And so with one molecule, you could say, well, longer KIR or higher KIR, maybe it doesn't matter, but as long as you have KIR from day 6 to day 11, you're going to get Calvert-Cesar cells. I think that's interesting, like to see this from a reductionist perspective. We also don't know, if you get the same cell, cluster of cells here through 2 different paths, whether they have maybe some difference in identity later on. That's something we can't really address. But there are definitely examples like that. And yeah, so and with each of these morphogens too, they can have the other thing just I want to bring up is that we are adding these morphogens exogenously, but endogenously, these as they're developing, they start developing their own organizer cell populations within them. So we know we're generating floor plate cells within our screen here. And that means after we stop adding whatever molecule we're adding, they're going to still be expressing sonic hedgehog within the organoid itself. And that's another layer of complexity we can't really well model just yet.
And
I hope I
Fatima,
answered your question. Yeah.
Fatima, do you want to jump in with a question? See your hand up there.
Yeah, thank you. So very cool, Neil. Thanks a lot. I was wondering, so your organoids are quite old. They're quite mature, and I was wondering, like you also find progenitors in them apart from neurons, right? And what would be the percentage of progenitors versus neurons in your organoids in general? Do you see like differences between different regions? Like, do you get, I don't know, more progenitors left over after a while from hindbrain or from forebrain, something like that?
Yeah, thanks for the question. I don't know off the top of my head. So yeah, indeed, we want to go for the 70-day time point because we felt we would get the most number of neurons, which we're most curious about. And we did get, I don't know the number off the top of my head, but maybe 50% or more neurons in the screen compared to non-neural glial cell types. Regarding the question of the proportion of progenitors in the different conditions, I think I'd have to check. I think in one of our supplemental figures, like I think 4 or 5, I think I can briefly check here. Here, I'll just share screen again here. Okay. So this is the breakdown of our conditions and the populations here. So we didn't really break it down by progenitor and neuron here, but you can definitely see it's varying quite a bit. And yeah, so I don't have any kind of grand summary of that off the top of my
Well,
head.
the green should have done it for you. I think you had progenitors and IPCs on there, right? The 2 green.
We have progenitor and IPCs, but then that didn't include the OPCs or the choroid plexus, which I guess the question was about progenitor, so you're right. Yeah. So there's and yeah, it did seem like, yeah, judging by that from the interneuron protocols had fewer progenitors, and the forebrain and Calabretia seemed to have more of those, I think. Yeah.
I see, thanks.
Any other questions?
Well,
Yeah.
maybe I can just, oh, hold on, we'll wait. I'll just wait. Yep.
Go for
Sorry,
it, Neal.
thank you very much. Quick question. I just wonder if you have looked at the sex hormone by any chance, testosterone, estrogen, apart from the morphogen, to see the impact, or is it in the pipeline by any chance?
We did not add anything exogenously in our screen that would impact hormones. So I don't know. I know I don't know off the top of my head what the results were of those papers, but I think there were a couple of papers in the last couple of years looking at that. Yeah, it would be really interesting to see. Yeah.
Thanks.
Maybe I can go back to this question about variability with you.
Sure.
So could you show that slide again where you showed a panel and you were comparing OLEG2 measured by the sequencing and with the antibody?
Yes.
It's a bit, yeah, there we go. So I was just trying to really grasp this. It seems very straightforward. So this is the percent of oligos in the culture. Is that what percent oligo lineage means? The percent of cells that are oligopositive. Olig, expressing an olig signature.
Yes, yeah. So, sorry, I cannot show here. Yeah, so for the olig lineage by single cell, that is any of the cells within the olig-2 progenitor or oligodendrocyte clusters. We had 3 clusters that were kind of various stages of maturation of oligodendrocytes.
But am I correct in thinking that this is the percentage of cells in an organoid or in some kind of unit of cellular? What does percent oligo lineage refer to?
Yeah, I can maybe show that better. Oops, maybe here. So it would be the number of cells from a given condition that land in this OPC mature cluster here. So like, for example, I think, so this is actually a mixture of a few clusters, but for a few conditions, but these are all the conditions that were treated with SAG, and you can see they generate a few different cell types, but maybe, in this case, in this grouping, maybe 30% land here. So that would be percentage there.
Great, thanks. So if you then go back to the image, it looks like for a given concentration of retinoic acid, you have almost like a kind of a linear response. I mean, it's like, is that how I should understand it? Retinoic acid 10 nanomolar is this top group of points here, and there's only 100 nanomolar.
Yeah. So maybe I should be moving this RA. So each of these dots is one condition from the screen. Many of the conditions in the screen you can see are 0, meaning they didn't generate any oligodendrocytes. And maybe like 10 or 15 or so generate some oligodendrocytes.
So these are all the different conditions.
That's right. Yeah.
Got it.
And so actually, most of these are, that's okay. Yeah. And then most of these had either some sonic hedgehog or retinoic acid in them that generated oligodendrocytes, and the RA 100 here. Yeah.
So that appears almost like linear then. I mean, across these conditions, you might say it's arbitrary, but you get oligodendrocytes across all of these conditions. And then earlier on in your presentation, you used the word label transfer in an interesting way that I thought might be worth going back to, because, I mean, is this just a Surat label transfer procedure, or because if you've got multi-omic data, so you have one set of data over here that identify oligodendrocytes, and then you want to do a label transfer experiment to ask which of my clusters are most similar, I suppose. You see what I'm getting at? It seems to me that you've got here, it looks almost like qualitative, that they're distinct colors, but in the other curve, it's kind of graded.
Yeah, this is a really important point around all these new technologies at single cell sequencing, as I kind of mentioned just really briefly earlier on, and that the annotation of cell populations is it's between an art and a science. It is becoming kind of like a skill set in its own, of like, how confident can you be in a cell population? Is it really the one you thought it was? And also we have to acknowledge just differences in platforms. Here we use parse. Other groups had used 10x. These are really different methods in quantifying, because 10x uses poly-T pull down of polyadenylated transcripts. Parse uses random hexamer priming. We have different levels of sequencing for our cell types. We have different timing. Ours were day 70 of differentiation. We're mapping to discrete stages of human development. And then also tissue handling, dissociation procedures. So all these things, I see it kind of as, it's quite remarkable we can do this at all, considering how different, the data can come from halfway around the world, and we can still somehow map them on with some level of confidence. But it is an art. And when we look at other papers, other groups look at our annotations, we want to be confident in what we're calling a population, so that others aren't misled, and that we're also just being as conservative and realistic as possible.
Oh yeah, am I still on here? Yeah, I think this is very cool. Because actually, although you look at it like this, it all looks qualitatively different. Actually, in some way, each of these is a representation of all the cell types. Doesn't that remind you a little bit of Yamanaka, for example? So yeah, it's a very interesting discussion. We could go on. Thanks very much.
Thanks for the question.
Perhaps connected to the same oligo observation, I was wondering, because you look at 70, the couple sonic hedgehog and retinoic acid treatment conditions had hugely increased numbers of oligos over the other conditions. I was wondering in my own head, I was guessing it wasn't a direct effect of those morphogens that created them, but rather the lineage that you get, maybe the ventral lineages that and posterior lineages that might be encouraged by those signals. Do they just move faster? So by day 70, you're getting oligodendrocytes where you would have to wait another month to get that in a dorsal lineage. And, you know, do you have a sense of either this effect in particular in oligos and other things you're seeing? How many of them are direct morphogen lineage induction, or once you make the choice of dorsal versus ventral or anterior versus posterior, things move at different speeds intrinsically in those regions of the brain?
Yeah, I mean, this is getting at the, you know, the levels at which we can't yet really comprehend how much are we kind of set a ball rolling down a hill and then the hill kind of takes its course, or are we kind of pushing it down all these paths manually? And because we're only you know, I think the best case is this cerebellar protocol where we're adding FGF2, which usually is basis of like a cortical protocol. But so it's basically the same until day 21, but then we add insulin through the N2 supplement after that. And that's enough from day 21 to day 70 or so to push it all complete, basically the cerebellum, a cerebellar fate versus a cortical fate. So I think these decisions are happening throughout, and we don't know when it, you know, maybe in that case, like we don't know if it's moving down a cortical fate and then it shifts to cerebellar fate, or maybe it's in this amorphous in between state and then it is deciding later. I think, you know, initially I want to do this screen because we could use these new technologies, and now after doing it, I think the next level of questions that you're asking and other people are asking really is going to come to like getting single cell barcoding of exposure to different morphogens, such as using like a GLI promoter driving like a CRISPR barcode system that whenever that cell receives sonic hedgehog, it'll, you know, encode something in its DNA. Whenever it receives wind signal, it'll encode that in its DNA. And I think these systems are still in development, but they can be used to record the experience of every single cell and what they experience, what signals they received at even specific time points. They can be temporal barcodes too. So it'd be amazing to really know, you know, you could pick out one cell and say, when was the sequence of Wnt activation, sonic hedgehog, everything else that it experienced. And I think that'll be possible, just not quite yet, but that'll be possible maybe next 5-10 years.
Did you guys look at any earlier time points than 70 with any of these?
Not in this
data?
screen.
Okay.
We did confirm things with immunohistochemistry, but nothing with single cell for this screen beforehand.
Gotcha.
Yeah.
Any more questions from the group? If there aren't any more, I can maybe show you just one or two things I have open Nemo to give you an idea of what we could create together to sort of start looking at all this data together. So this is a Nemo web page, again, open, functional right now, and I'll send out links again. This is the La Mano data. I've just asked to look at the FGF17 gene, where this is a little bit small to see, but this is sort of the whole study. And here, each of these windows is broken down by region. So these are the dorsal forebrain dissections across all the ages. And you can see there's a very small population of FGF17 expressing cells here. And FGF17 is associated with the rostral patterning center in the telencephalic development. And here it's broken down by cell type, but you can, in Nemo, you can click this button here and look at different displays to look at things other ways. And the other way I looked at it was by age here. So this little population that lights up with FGF17 is blue, which is the earliest time point they measured. So you can see, and this area here is the sort of large population of radioglial cells that then move through IPCs over here into neurons. And sort of and this is sort of important to think about how these organizers work. They're very small cell populations that have very huge impacts. So in a very small population at a very early particular time, FGF17 is turning on to tell cells where they are and what they're supposed to be doing. And presumably, in all of the complex experiments you just described, the different combinations of these organizers are appearing and disappearing and signaling to create all those different fates that you've observed. And this is just to show that we sort of have in our hands, we collectively, by getting these data together, have these answers or these resources to get some answers. Here's the same gene in the macaque data. So this is the Mikali study from the end of last year. And you see, again, a tiny little population of FGF17 expressing cells, again, presumably at early times. And in this paper, Nicola really did a sort of exquisitely precise dissection of the, not physical dissection, but informatic dissection of the patterning centers, where they actually had an entire analysis dedicated just to them, where these are all different organizer identities. And you can see here, this FGF17 lighting up the purple class here, which they identified as the rostral patterning center already. So it's sort of a circular confirmation of what's going on here. But again, this is just to kind of show you what's available on NEMO, what we can see when we get these together. And if we can bring these in vivo resources in the same space as the incredible morphogen studies so many of you are producing, it'll really just be a fun ride. And just one final note, in addition to looking at these individual genes, we have enabled in NEMO Analytics the exploration of these same data sets by gene lists or weighted gene signatures. So if you have a principal component or a cluster of genes or a dimension from NMF that you have gene weights for, you can similarly visualize it across all of these data resources, just like we're looking at individual genes. And all you have to do is upload a gene list. So I'll be contacting a lot of you to get sort of some momentum and to entice you into this data environment. But it's really incredible. If you bring those two things together, A is tons of data, and then B is either genes, gene signatures, or specific transcriptional dimensions that are of interest, whether they came from a patterning center or a particular lineage, you can really start to see expansive, very powerful perspectives on all of this incredible data that the field is generating. So just wanted to kind of give you a glimpse there. Neal, thanks so much for going over all that data with us. And next month, as I said, we have another set of talks on organoids and morphogen control. So looking forward to more of this, and hopefully I'll have some of the data from these talks up and we can start to look at it together if you're interested in that. So thanks to everybody, and Neal, great to meet you today. Thanks all.